Today, we're diving into a detective story, but this one happens on a scale so small you can't even imagine it. We're talking about the case of the Radioactive Spy.
So, think about this for a second. When you take any medicine, how do scientists actually know it's going to hit the right spot in your body? How can they be sure it's going to work where it's supposed to and only where it's supposed to?
Well, it's a huge question, and the answer isn't simple. It's actually a kind of detective story, a really tiny one, that plays out in labs all over the world.
And our story starts with what we'll call the lock and key mystery. This is the fundamental challenge, right? Finding that perfect 1 in a million match between a drug and its target inside our cells. Okay, here's the best way to picture it. Your body is filled with these proteins on your cells called receptors. Think of them as tiny, unique locks. The D3 dopamine receptor is a great example. Each one of these locks does a very specific job. Now, the drugs that scientists create, those are the potential keys. For a drug to work correctly, its key has to fit one specific lock perfectly. If the key is a bad fit, or if it fits the wrong lock, well, that's where you get side effects. Getting this right is absolutely everything.
So, how do our scientific detectives actually solve this mystery? Well, like any good investigator, they need the right tools. So let's meet the target of our investigation and their high-tech informant.
First things first, every detective needs a crime scene. In this case, scientists have to create it. They take cells, literally break them open, that's a process called lysis, and then they spin everything in this super fast machine called a centrifuge. This separates out just the cell membranes, which is the part where all those receptor locks are located. It's all about clearing away the clutter to focus on the clues.
And this is where our informant comes in. Every detective needs a spy on the inside, right? Meet the radioactive spy. In our story, it's a molecule called 3H-spiperone, and it's designed to do two very important things. One, it is built to stick to our target, that D3 receptor. And two, it's tagged with a little bit of radioactivity, which works just like a tracking beacon. It's the perfect spy to send in to find all the locks.
All right, the scene's prepped, the spy is ready to go, it's time for the investigation. Let's walk through how these scientists actually gather the clues from this tiny, invisible world.
The whole procedure is called a radioligand binding assay, but it really boils down to 4 key steps. First, they prep the scene, putting those isolated membranes into a tube. Step 2, they send in the spy. The radioactive spiperone gets added in, and they just let it sit, for about 75 minutes in this case, so it has time to find and stick to all the receptors. Step 3, capture the evidence. They pour the entire mixture through a special filter that catches the membranes and our spy along with them. And finally, Step 4, count the clues. A machine called a scintillation counter measures all the radioactivity, which tells them exactly how many spies got stuck.
Okay, that all sounds pretty neat, but wait a second. There's a catch, isn't there? How do we really know our spy is binding to the actual target and not just randomly sticking to other junk on the membrane?
This is a huge deal. It's where we have to separate the real clues from just random noise. You've got specific binding. That's our spy fitting perfectly into that D3 receptor lock. That's the signal, the clue we're after. But then there's non-specific binding. Think of it like static on a radio, or just dust sticking to a surface. It's meaningless noise, and if you don't account for it, it can completely ruin the investigation. So how do they filter out that noise? It's actually pretty clever. They run a second parallel experiment. They add what's called a blocker molecule. One example is plus butaclamyl. This thing rushes in and plugs up all the real D3 receptor locks first. Then they add the radioactive spy. So any radioactivity they measure this time has to be that random, nonspecific noise because all the real binding sites are already taken. They just subtract that noise number from their main result, and voila, they have a clean signal. Okay, the clues have been collected. The noise has been filtered out. Now it's time to analyze the evidence. What does all this data actually tell our detectives? So the very first clue they uncover answers a really basic but important question. Just how many of these D3 receptor locks are there to begin with? This value is called Bmax, which stands for maximal binding. It literally just tells them the total number of available receptors on those cell membranes. It's like doing an inventory of all the locks at the crime scene. It's critical for understanding just how big their target is. All right, next up is clue number 2. It's not enough to know how many locks there are. The detectives have to know how well the key, our spy, actually fits into them. This is where a value called KD, or the dissociation constant, comes in. Now listen close, because this part is a little bit backward. You want a low KD value. A low KD means a super tight, super sticky bond. It means the key fits the lock perfectly and it does not want to let go. A high KD means a loose, sloppy fit. So remember, for KD, the lower the number, the better the fit.
And that brings us to the last, and maybe the most important clue of all. The whole reason we're doing this is to test new drugs. So how well do our new suspects compete against our trusted spy for a spot in that lock? This is what the Ki, or inhibitory constant, tells us. You can think of this like a microscopic competition. The scientists throw in a potential new drug to see if it can push our radioactive spy out of the way and take the receptor for itself. The Ki value tells them exactly how good that new drug suspect is at doing that. It's how they rank all their potential candidates and figure out which ones are worth pursuing.
And this chart just lays it all out for us. It's super simple. Just like with KT, a lower Ki number is what you're looking for. See drug A? It has a low Ki. That means it's a really strong competitor. It binds tightly to the target. It's a prime suspect. Then you have drug B with a high Ki. It's weak. It can't really compete. This tells scientists, okay, forget drug B, let's put all our energy into developing drug A.
So, we've followed the clues, we've analyzed the evidence. Let's take a step back and connect the dots. How does all this microscopic detective work on a lab bench lead to the medicines we use every day? Why does it matter so much?
Well, this one technique is basically the bedrock of modern drug discovery. It lets scientists sort through literally thousands of potential drugs to find the ones that have a real shot. It's how they weed out the molecules that would cause all those nasty side effects. It even helps them figure out the right dosages. In the end, it's what helps us get safer, better medicines way faster.
So, the main thing to remember here is that this isn't just some obscure lab experiment. It's one of the most fundamental, most critical steps in the entire process of designing safer, more effective treatments for all kinds of diseases. You know, this radioactive spy helped solve one tiny but incredibly important mystery. It really makes you wonder, doesn't it? What other invisible puzzles are scientists solving right now that are going to completely shape our future?